Hi, so I'm Seyler. I am just beginning my second year as a postdoc at Michigan State University, working with Matt Schrenk. And I'd like to talk to you today about using metabolomics to look at metabolic processes in serpentinizing environments. I'm very excited to be kicking off the ROC session and also to be introducing you to serpentinizing environments. A couple of people in this session are going to be talking about them. So it's pretty cool that I get to introduce the topic to you if you're not very familiar with it. So, serpentinization is the process by which this really pretty green rock turns into this really ugly black rock. Basically what that means is that the mineral olivine reacts with water and produces serpentine minerals as well as high amounts of hydrogen, so it creates a very high pH environment, energy in the form of methane, and also these small organics which can be food for microorganisms. So it creates this really incredible environment where it's very extreme. There's very low amount of dissolved inorganic carbon available because it all precipitates as calcite, and it's very high pH. But there's all these organics and there's all this energy available for microbes to harness. So it's a very extreme environment, but with a lot of opportunities for microbes. Also, it is interesting because not only was it very prevalent on early Earth, but it's also very likely that serpentinizing environments exist in the subsurface of Mars and fuel hydrothermal environments on icy worlds like Europa and Enceladus. And there are a number of origin of life theories that implicate serpentinizing environments because they create these high energy environments with gradients that can be harnessed by early life. So there are a number of different serpentinizing environments all over the globe that our lab studies, but I'm going to focus on the Coast Range Ophiolite Microbial Observatory. It's a nice place to work because it's just north of Napa. So, you know, drunk science. But anyway, there's two sites that we work at here at Chromo, the Quarry Valley and the Coreshed Well. And I'm going to be talking about some microbes isolated from Quarry Valley. We have 6 wells dug at Quarry Valley and another 6 dug at the Coreshed Well. And they are highly alkaline. And that's the drill rig that was used to drill these wells just a few years ago. So we have a ton of 16S data from these wells. We have a pretty good idea of what kind of microbes are living in these highly alkaline environments. And as you might suspect, they're not very diverse because it is a very extreme environment. But we do see some trends associated with pH. Certain groups show up in the higher pH wells versus the lower pH wells. I'm not going to focus on any of that today at all. I'm not interested in who's there. I'm interested in what they're doing. And in an environment like this, it's very difficult to distinguish between biotic and abiotic processes. These reactions occurring between minerals and water producing organics, producing methane. Microbes produce organics and methane. So it's difficult for us to figure out what's going on at the geological level and what's going on at the biological level. And it's very likely that the microbes are feeding off of these geologic processes. So there's a very gray area here. So that's where a technique like metabolomics comes into play. Metabolomics basically is a snapshot of the metabolic activity of a microbe. It takes all of the small organics that microbes are producing and looks at them at the chemical level to try to figure out what metabolic processes are occurring in the cell. And this technique can be used for a number of different applications. We can figure out, whoops, I got ahead of myself. We can figure out what chemicals are being produced by the cells, obviously. We can annotate enzymes based on what is being produced. We can look at pathways. We can figure out what metabolic processes are occurring. We can even phenotype different cells that we can't, different organisms that we can't necessarily culture in the lab. So this is all very useful, right? But there are some caveats, of course, just like with every technique. First of all, no extraction method is ideal. So especially if you're doing an untargeted approach and you want to look at all of the metabolites being produced by microbes. Now, no extraction method is going to get every single metabolite. There's always going to be some bias involved. There's a bunch of different ways to analyze the metabolites once you've extracted them. We use liquid chromatography mass spec, but you can also use GC, you can use NMR, and each one of those methods has its own perks and drawbacks, which I'm not going to get into right now. So the way that we characterize each of these compounds once we've extracted them is looking at the mass to charge ratio and the retention time in the column. But that can vary depending on your methodology, right? So if you're trying to characterize a compound based on these things and it varies from method to method, then one person will get completely different results from yours in another lab. It only provides potential chemical formulae, and this is especially a problem if you're looking at really big molecules, because you get these results where you have, like, okay, it can have so many carbon atoms in it, so many nitrogen atoms in it, so many oxygen atoms. The bigger a molecule is, the more potential possibilities there are for that molecule's identity, up to thousands of potential identities, right? Stable isotope labeling can help constrain that. So if you feed microbes with, say, 13C, that will help you constrain at least how many carbon atoms are in the molecule, which narrows things down quite a bit. And obviously, we haven't identified every single metabolite that exists. There's a lot of them. And there's a lot of what we call unknown unknowns. We don't know that it's there, and we don't know what it is. So that's also an issue. But databases are currently being developed, and they're constantly being added to, and work like this helps enhance the development of these databases. So a technique like this is a lot more powerful when you combine it with other molecular techniques like genomics or transcriptomics. If you know what genes microbes have or what genes they currently have turned on, then you can kind of guess at least what metabolic processes are occurring and then use that to inform your identification of all the metabolites. And vice versa. If you see certain metabolites that are being produced, you can say, okay, well, I know certain metabolic processes are currently occurring. So metabolomics can also help you annotate a genome or a transcriptome. So this is what the data looks like coming out the other side of the LC-MS. We use tandem liquid chromatography mass spec combined with quadrupole time of flight. If you don't know what that is, don't worry about it, but this is what the data looks like. And each one of these peaks represents an individual metabolite that we can pull out and try to identify. It's not very informative to look at this though, right? So this is a tree of a number of isolates that we have from chromo. And I know every time I see a phylogenetic tree in a talk, my eyes start to glaze over because this is a lot of information and it doesn't really mean much. But I want you to focus on these two isolates over here that were isolated from Quarry Valley 1-1 well, which has a pH of about 11.5. And the reason why it's important is because they're both very closely related to each other, but they're also closely related to Bacillus pseudoformis, which is a very well-known alkalophile that's pretty well characterized. So we decided to focus on these two isolates for now because they're so closely related to a microbe that's genome has been sequenced that we know quite a bit about and what its preferences are when we're trying to grow it in the lab. So I grew these two isolates, and as you can see by the growth curves, it's a pretty standard growth curve. We have your exponential phase here where the cells are very actively growing and dividing, and then they just kind of plateau off here at the stationary phase where you have the same number of cells being produced as are dying. So what's interesting here is that the number 1 isolate takes off pretty rapidly, but then number 2, even though it's very closely related to number 1, has kind of a different growth pattern here. So it has this little dip in the beginning, and then it goes into exponential phase. So we decided to look at the exponential phase and the stationary phase of both of these isolates and compare the metabolites being produced by both of these isolates to see if they're using different strategies to adapt to this highly alkaline environment. And what we found is that this is a PCA plot of the total metabolites of each one of these cultures. And you can see that the stationary phase of both of these isolates clumps pretty closely together, but they are distinct from each other. Likewise for the exponential phase. And then our blank is all the way over here, which is always nice. So we know we have good, clean controls here. So the cool thing is that we can look at individual peaks and look at their relative abundance between all of our different samples. And you can see that there's certain compounds that are present only in the exponential phase of both of these isolates or only in the stationary phase. And then there's ones that show up only, for example, in the number 2 culture, but not in number 1 at either phase of growth. So that's kind of neat. And then there's also compounds that only show up during one phase of growth in one isolate. So all of these compounds are distinguishing between these different isolates at their different phases of growth, and we can use them to look at what metabolic strategies are being used in these isolates. We haven't identified these compounds yet, and that's going to be an interesting exercise because, again, they're very likely unknown unknowns. If they were very well-known metabolites, they'd probably be seen in all of the cells because they're extremely common and very readily identified. So we're going to start searching the databases and see what we can come up with. But we also have transcriptomic data that we need to analyze from these cultures, and so we can combine that with the metabolomics data to try to get at exactly what strategy these isolates are using to survive in this extremely alkaline environment. The thing about these bacillus cultures, though, is that they're not really going to tell us a whole lot about what goes on in a serpentinizing environment because they're aerophilic. And because bacillus is a pretty common organism and it's found basically all over the place. So we can start to understand things about like alkali tolerance, tolerance of high pH, but understanding some of the other processes that occur in serpentinizing environments, looking at the bacillus is not going to be particularly useful. So we have this isolate, Methanobacterium subterraneum, which is not from a serpentinizing environment, but it's very closely related to organisms that we have identified in serpentinizing environments, including chromo. This was actually isolated from a subterranean aquifer, I think in Sweden in 1998. It is alkalophilic, just like the bacillus cultures, and halotolerant. It will grow at a wide range of temperatures, but it's also a methanogen. And it might convert formate, CO2, or both into methane. So we started growing this in the lab anaerobically. There's a couple of different carbon sources available in the medium. I focused on formate and bicarbonate because they're implicated in the production of methane. And I added a 13C label, bicarbonate and formate. So I set up cultures where either the bicarbonate or the formate was labeled with C13 in the hopes of tracking that into the metabolites. And these were harvested during the exponential phase. I put a question mark here, whoops, because I'm not really sure if this was the exponential phase. We haven't established a very good growth curve for these because they clump a lot and it makes it difficult to do cell counts. Unfortunately, we didn't get as neat of a distribution here. So the media looks an awful lot like blank media, which means that if these cells are producing extracellular metabolites, they're not as easily distinguished from the rest of the media. And also, we couldn't really tell much of a difference between the cultures that were fed 13C labeled formate and the cultures that were fed 13C labeled acetate. And this makes sense if you look at the peaks, because if 13C label was being incorporated into the metabolites, you would see a distribution of various peaks here, like a spread, where there's compounds that have incorporated some of the 13C, and there's ones that still have 12C. It doesn't look like any label has been incorporated here. So all of the 13C is probably going into the methane and not into the metabolites, or at least most of it. But again, we can still see there's certain compounds that occur only in, say, the pellet or only in the media. So our next steps are to grow the bacillus in more defined media, sequence the genome, and also look at the transcriptome to try to tease apart what exactly is going on metabolically in those isolates. But also to take the Methanobacterium and try feeding it 13C acetate to see if that is incorporated into the metabolites, trace the 13C into the headspace gases to see if it's going into the methane from the older cultures. It should be, it's gotta be going somewhere. And also look at the transcriptome and hopefully develop a better growth curve. We've been looking at protein assays as a substitute for doing direct cell counts. And we're also working on doing field metabolomics on environmental samples from chromo. So with that, I want to thank you for your attention, and I'll take your questions.
Questions for Lauren?
Yeah. Had a feeling you'd have one, Nadia.
So I saw that you have abundant Comamonas and rhodocyclasia at the beginning when you sampled and looked for the 16S. Mm-hmm. Did you also get to isolate those, and in what type of media?
In which? In some of the earlier, the 16S
One of the
data?
first slides you showed were all the 16S abundance patterns.
Take a minute.
There.
Oh, there. Okay.
You have, so pretty much the red and
Yeah.
Oregon.
So I believe some of these have been isolated. I don't know what media we're currently growing them in, so I've been focusing on the bacillus. But if you look at the tree, we have a ton of isolates right now that we just haven't actively cultured because there's just so many of them. But that's in the works. Yeah.
Okay, let's thank Lauren one more time.
Thanks.